Hello student, welcome to the lecture. In this lecture we are going to discuss the metalloenzyme of copper, that is plastocyanin. In previous lecture we discussed the metalloenzyme of copper, that is blue oxidases. Now in this lecture we are going to discuss the structure, function, reaction and biochemistry of plastocyanin. So let's start with the plastocyanin. Plastocyanin, as we know that the plastocyanin is a blue copper protein. It is found in plants, which play a very important role in photosynthesis. It is an intensely blue copper protein. So let's discuss the structure of plastocyanin. As we see in the figure there, the copper is at an active site, which is 2 sulfur and 2 nitrogens. These are the only binding site atoms are written here. So this 2 sulfur, one is from the cysteine residue and the second sulfur is from the methionine residue. Here the 2 nitrogen is from the histidine groups. So here this is an exact structure in which the ligands are clearly shown in that figure. So these are the same one. Here again the copper is an active site. Here 2 nitrogen, this is the histidine group of the 2 histidine groups act as a ligand, and the second sulfur is from the methionine residue and the next one is from the cysteine residues. So the copper is an active site in the 4 ligands. In that there is a 2 histidine nitrogen atoms are attached to copper and 1 sulfur from methionine and cysteines are attached to the copper. So let's discuss the structure in detail. So in structure we can say that actually plastocyanin is a first metalloprotein which is characterized by extracrystallography. So 8-stranded anti-parallel beta-barrel structure with 1 binding site, that is copper is at active site. So here let's discuss the structure in detail. As we know that the plastocyanin is blue copper protein. Plastocyanin, in that the copper is an active site which is ligated by the copper is bind the 2 histidine residues. In that the next one, that is the third and fourth position is occupied by that is 1 sulfur from methionine and second sulfur from the cysteine residue. So the sulfur residue from methionine and sulfur from cysteine residue. As we see in structure that the structure or the geometry of this metalloenzyme is distorted, trigonal, pyramidal. So the geometry of is distorted, trigonal, pyramidal. Now here if you observe that the 2 sulfur, one is from methionine and second one is from the cysteine residues, the 1 sulfur contact is shorter than the other. Due to this elongated sulfur contact, the binding or the oxidizing power of the copper is getting increases, and the intense blue color of this plastocyanin is due to chance transfer transitions. Actually, in plastocyanin, the hydrophobic patches are present. Due to these hydrophobic patches, the binding power of this metalloenzyme getting increases. Plastocyanins also present in hydrophobic patches. These hydrophobic patches are surrounding the exposed histidine to bind the copper sites. The acidic sites or the hydrophobic patches are present in many plastocyanins. Due to these hydrophobic patches or the acidic sites, there is an increase the chances for binding of another metalloenzyme which is help in the electron transfer reaction. So we can say that the hydrophobic patches or the acidic sites are present in plastocyanins. Due to this, the recognition or the binding sites the binding sites of another metalloproteins or the metalloenzyme, which helps in electron transfer reaction. This increases. So these are the overall structure of plastocyanin. Now let's discuss what are the function of plastocyanin. So let's discuss this function of plastocyanin. As we know that the plastocyanin is a metalloprotein or the copper binding protein. In that, the copper is an active site. Plastocyanin transfer electron in plants. Actually, it is present in a plant. So let's discuss the function. As we know that the plastocyanin transfer electron from cytochrome F to cytochrome B6F. Then the second function is that cytochrome B6F is from the photosystem first. We know that photosystem has two types of system, photosystem first and photosystem second. In photosystem first, the cytochrome B6X is present in photosystem first, and that is plastocyanin, that is 700 is in solid band, is from photosystem second. Then we know that the third function is that the cytochrome B6F complex and that is solid band again are these two proteins, means that cytochrome B6F and plastocyanin 700 plus are both membrane bound proteins with exposed residue on the lumen side of thylakoid membrane. Thylakoid membrane of chloroplast. As we know that the photosystems are important reactions in synthesis of plant. So here synthesis of proteins in plants. So actually here this photosystem take place in the chloroplast. So in that the electron transfer is from cytochrome F to cytochrome B6F. So here the cytochrome F is act as a electron donor. While act as a accepts electron from reduced plastocyanin. So these are the important functions of the plastocyanin. Now let's discuss the main reactions of plastocyanins. These reactions are similar with their functions, as we know that the plastocyanin is electron transfer protein in plants. So let's discuss the reactions. As we know that the function, the plastocyanin can transfer electron in plant. So here in the reaction we discuss the exact reaction of this plastocyanin. So in reaction plastocyanin is get reduced by the cytochrome F, and the reaction is as like plastocyanin is reduced by cytochrome F. Then here the electrons are added. So the reaction is as follows. Here PC means this is a plastocyanin. It is a short form used over here. Then in added electrons, so the reaction is as that. So here, as we know that the after dissociation of this plastocyanin, reduced oxidized plastocyanin, dissociation of, actually what happened after dissociation, this plastocyanin, oxidized plastocyanin, diffuses through the lumen site, until recognition or binding. Recognition means binding. Binding occurs with that is plastocyanin 700 solid band is get oxidized, and the reaction is as follows. At which point this plastocyanin is oxidized, and according to that the reaction is as follows. Here again the same reverse reactions are happen over here, that is so the overall reaction is as like that. In that first the reaction, the plastocyanin is reduced by cytochrome F, and the reaction which is taken place is as like. So here again, after dissociation of this copper plastocyanins, means oxidized plastocyanin, this diffuse through the lumen site until the recognition of the binding occurs at the point which oxidized reactions, that is at this point the plastocyanin 700 that is oxidized, and the reaction is exactly opposite to each other. Now the plastocyanin is reduced by the cytochrome F, and the exact reaction is take place as like that. Now this oxidized plastocyanin is diffuses through the active site until the recognition or the binding sites occur through the plastocyanin 700 band, at which point it is oxidized and the reactions is taken place which is exactly opposite to the first one here again. Now the redox potential for this reaction is a 340 MV. So the redox potential is MV, and isoelectronic point is 4. So these are the important physical parameters for this reactions which is done by the plastocyanin in chloroplast. So these are the overall structure, function and reactions of plastocyanin. In next lecture we are going to discuss the next metalloenzyme of copper, that is tyrosinase. Thank you.